Hello, welcome to Convergent Dialogues. This is Xavier Bonilla. On this episode, I am talking with Raghuveer Parthasarathy. He is a physics professor at the University of Oregon. He's been there since 2006. He has a PhD in physics. His research interests include biophysics, physical properties of lipid membranes, and the assembly of multicellular structures, such as in embryos, and looking at all the different ways of using optic microscopy. He is the author of the new book, So Simple a Beginning: How Four Physical Principles Shape Our Living World, which is fantastic. It is a fabulous book. I highly recommend it to everyone. And that is the bulk of what we talk about in this conversation. If I recall, this is his first book, and so we talk about why he decided to write this book for, kind of a popular science audience. We discuss what is biophysical. It's kind of this merging of two worlds, and he gives a good definition of what that is. He talks about why he chose the four physical laws that are discussed in the book. There are other physical laws, but he gives his rationale for why he chose the four that he chose. We talk about the mechanics of self-assembly, the interactions of regulatory circuits. We talk about the Brownian notion, predictive randomness. We talk about scaling and the role of metabolic rate. We give an overview of genes and how genes work. We talk about sheaths and helices of genes and their importance, histones and self-assembly, lipids and the gut microbiome, and then we end with how to read DNA and some of the ethical questions that people have around CRISPR. I have to say that he was such a wonderful person to talk to. When you listen to the conversation, you'll be able to hear that he's a very good teacher. In many ways, he's very good at explaining very, very complicated and complex things. And the reason why, I can recommend his book so highly is because he does just that. He's very good at explaining very difficult concepts, I think, for, the layperson that's, not within the physical sciences, in understandable ways. And so, I've been reading and talking to many people that have been talking about genes and biology, and he's up there with one of the best in how to explain it. And so his book is very good, and we have a wonderful conversation about his book and many of these principles that are in the physical world that are important for us and many organisms, and really gives the kind of defense of why we should study this stuff and know about it and know it for ourselves and for the world around us. So now I bring you Raghuveer Parthasarathy. I'm here with Raghuveer Parthasarathy. I'm hoping I got that close. How are you?
Great. Yeah, it's great to be here, and thanks for having me.
Absolutely. I'm really excited to talk with you about your book. The title is So Simple a Beginning: How Four Physical Principles Shape Our Living World. And it's a fabulous book. And so I definitely want to talk about it. Before we get into it, just tell us who you are, what you do, what your research is in, and all of that wonderful stuff.
Yeah. So, I'm Raghuveer Parthasarathy. I'm a physics professor at the University of Oregon. And for many years now, I've been exploring biophysics, which I'm sure we'll talk about how to define that, which is actually a very challenging thing in itself, how to define what biophysics is. But anyway, I'm a biophysicist, and my research group especially looks at things like gut microbes, but looking at them kind of from a physical perspective of how they aggregate and move and swim and things like that. And my lab has also explored things like kind of the material properties of cell membranes and how they can organize themselves and build structures and do physical things to make themselves work. So I've been here a physics professor at Oregon for 15 years. So I started in 2006. And interestingly, even though at the moment, I'm very much kind of in both camps of being in physics and a physicist and a biologist. My official training is all in physics. My undergraduate degree was in physics from the University of California at Berkeley, and my PhD was also in physics. And after that, I spent 4 years masquerading as a chemist again at UC Berkeley. I was a postdoctoral fellow in the chemistry department there. And it's actually there that I really started getting into biophysics, starting to look at cell membranes in that work. So biophysics is also kind of an interesting topic because, it's, as you can tell from the name, the sort of intersection of biology and physics. And so people come to it from lots of different paths. And my path has been sort of more of a training in physics and then branching out into how this can intersect with biology. People come at it from a lot of different ways.
Yeah, it's fabulous. It's probably safe to say that you know a little bit about the physical sciences, just a little bit, just a little bit. You've been in all these different subsets, biology and chemistry and physics. So that's super fascinating. And I think that's good, right? That's where the field is moving. We're trying to, or fields, this multidisciplinary thing instead of people kind of, sanctioned off into their own, kind of corner. So that's what we need. That's good.
Yeah, and
I guess the first question I have is, I don't know if this is your first book or not. I guess for like a popular general audience, and why did you decide to write the book for?
Kind of general science? It is my first book, and I wrote it because this field of biophysics is a very vibrant one, very exciting. A lot is going on, and there's a lot of just fascinating stuff in it. And it really does merge kind of the variety of biology with kind of the principles of physics. But I've really felt for many years that it's not something that comes up a lot in like the popular science media. You hear a lot about dinosaurs and string theory and things like that, but like biophysics is kind of not on the radar of a lot of people, despite being really fascinating. So I've thought that for a while, and I also thought that comes up also in things like college education. And I forget when I first did this, but I actually decided to create a new course for non-science major undergrads called The Physics of Life, which is basically about biophysics and really conveying that, without doing too much physics or too much biology, if you combine these two, you can get some really neat insights into how the living world works. So my kind of main teaching interest in general is courses for non-science majors, so science literacy and that sort of thing. And I'm actually a co-director of our science literacy program here at the University of Oregon. So I'm really keen on reaching non-scientists in general. And I thought, biophysics, it's fascinating. And also it's a very good route to science literacy because it really does illustrate how different kind of principles apply across fields and how a lot of this intersects, our daily lives and like how, our bodies work and health and that kind of thing. So the course, I've taught that a few times and I've really enjoyed that. And, new courses are always a lot of work. And this was especially a lot of work because there isn't a lot of I had to kind of write a lot of materials and stuff myself for it. And, as I was doing that, I was thinking, it would be good to think more about a real popular science book on this. So I started kind of dabbling in that and kind of writing a few chapters. And then starting a few years ago, I decided to get really serious about that. And that was sort of the nucleus of it all.
Well, I'm glad you wrote it. It's really important. This again, I'm in the other side of things. I do mostly the social sciences, but there is this kind of overlap that occurs usually with evolutionary psychology, and there's obviously some elements of biology we learn in. So it is really, really important. I'll say the one really nice thing about the book is that it is pretty accessible for folks, and this stuff is not easy to explain, and you do a pretty stellar job. I was reading it and I was like, yeah, I can see how he definitely teaches, right? He's been teaching for a while. If you're able to explain this in very simple ways, usually that's a good sign of a good teacher. So it was great. It was very readable.
Well, thank you.
And so I think even folks that just have like kind of a high school, biology or physics or chemistry can kind of get it. So
yeah, or even not. I mean, my goal was to really make it very accessible but also very kind of contemporary. And so people who do know something, I hope will still get a lot out of it because I tried to put a lot of very contemporary things in it. But I really wanted to make a
Yeah.
Very minimal prerequisites in a way.
Yeah, no, that's wonderful. So, okay, so let's start with, you've already mentioned it, biophysical, right? What is this? How do you define it? And how does this help us understand this natural world? And it's kind of like this new field of sorts, or a subset of a field that's been occurring for the past couple of years.
Yeah, so that's a great question. Yeah, like what is biophysics, and how do we define it, and so on. And it's interesting to maybe first take a step back and like think about what we mean by physics.
Mm-hmm.
So, what is it that ties together our understanding of how planets move around the sun, how lasers emit light, how an airplane wing gets lift, how ice cubes melt in your drinks. These are all like really very, very diverse phenomena. But what we've realized is that they're all tied together by basically universal laws of electromagnetism, mechanics, gravity, and universal meaning that they apply to everything.
Mm-hmm.
And kind of flipping this around, that's almost what the definition of physics is. Like, what are the kind of commonalities, the universal principles that tie a lot of very, very disparate things together? And there's this realization that we've had now for a few centuries, but it's still really very striking that there are universal laws. It's the same law of gravity that affects two stars as affects two strawberries. Their masses and stuff may be different, but it's the same underlying rule. So you kind of think of physics as this the study of what is kind of general or universal, and you can apply that to very specific systems, and you have like liquid water boiling into water vapor, and liquid mercury boiling into mercury vapor, and anything that undergoes some kind of boiling transition might have different temperatures at which it happens, it might look different or whatever, but they all undergo this sort of same transition, let's say, from like a liquid to a gas. And it turns out if you look kind of at the molecular level, they're all doing this exact same sort of rearrangements at that transition. So you can have specific details, but there is still some underlying commonality to it all. And that's sort of the physics of it. So this, like we said, that applies to kind of everything. So it should apply to like the living world as well. I slip on a banana peel, and it's not that I turn on my gravity processing gene. It's that, it doesn't matter that I'm a living thing. I'm going to fall just like anything else. Right. So in a way, we can think of biophysics as how do the sort of general principles of physics, whether they're electrical interactions or mechanical forces or stuff like that, how do these manifest themselves in living systems? Mm-hmm. So how can we make sense of various aspects of the living world, either the structure of animals or the way signals go through neurons and stuff like that? To what extent is that a reflection of kind of the underlying sort of universal physical principles? And the hope, and this has turned out to be really quite true, is that there are a lot of things that thinking about that kind of physical perspective or that general perspective really does illuminate why things work the way they do. And it's not so much the exact molecular details, but kind of the underlying phenomena.
All of that is really important, I think, for people to understand really the convergence of things in our world, right? How do we take the laws of physics that we understand and apply it to things within our biology, or just biology in general? And now the next question that comes out of this is the subtitle of the book is 4 physical principles, right? Now, is it just your 4 favorites? Is there only 4, or why 4? I'll list them, make sure I have them right. Is self-assembly, right, regulatory circuit, predictable, randomness, and scaling, which is kind of had some time in the sun with the work by Geoffrey West.
Right.
So. These are the 4. So kind of explain, are there other basic principles, or if there are, why did you choose these 4? Are there only these 4? Kind of just talk us generally.
Oh, that's a great question. Yeah. So there aren't only these 4. And I should say that it's not some canonical set of 4 principles. These are really 4 things that I felt help unify things. And so different biophysicists would probably disagree. Well, actually, I have this conversation with people. I think there's a pretty good agreement that these 4 are important, but how people phrase them might disagree, and how people phrase them, different people will phrase them different ways, and there may be different things some people would add or subtract from this list. But yeah, it's not a canonical list, but it's things that I felt are sort of key to this unification that you can make between lots of very different biological phenomena. And one could certainly expand that list. It would be a bit tough to shrink it. One could certainly expand that list, but I feel like this is a good list. But it does, in a way, highlight. I was just saying that there isn't a canonical set of what principles are important. In a way, that highlights how vibrant the field is. In a way, it hasn't sort of calcified. Here's the core dogma of biophysics. We're still actually trying to figure out what are the best ways to think about all this.
So it's interesting because it's much like many things, right? People will, kind of debate and discuss, what are the kind of basic kind of structures of things, and some people may say it differently or frame it differently. So I guess the one of the first ones you talk about them kind of throughout the book, right, as you go. But one of the first ones that you spend time on in the beginning is this idea of self-assembly. So
yes.
What are the physical properties within biology that allow organisms to self-assemble? And I don't recall if you explicitly use this term, but where do you find the gene's eye view of evolution fitting in with self-assembly?
Oh, yeah. So self-assembly is, I think, one of the most kind of dramatic, but also very deep principles underlying a lot of things. So first, just to step back, what do we mean by self-assembly? Well, it's this idea that the rules for putting pieces together are kind of embedded in the pieces themselves. Or to give a concrete example, you take a soap bubble, you blow a soap bubble like we've all done many times, and that soap bubble, it's a beautiful sphere. And you can ask, okay, why is it a sphere? There isn't any complex chemical reaction that had to take place to turn it into a sphere. It's not even alive, so there aren't any genes that make it a sphere. Right. But the sphere, it turns out that the soap bubble is a liquid, and any liquid will minimize its surface area, and the sphere is the shape that does that. So the fact that it's a liquid, kind of embedded in that nature of being a liquid, is this mechanism of minimizing your surface, and so that lets it assemble itself into a sphere. So to take a slightly more complex example, that's actually one that I start off with in the introduction of the book. Again, if you play around with soap bubbles, you can blow them and sort of get them to settle on a surface, like baking sheet or something like that. And if you have soap bubbles sort of coming together, you'll have very particular kind of junctions between them. You'll never find, for example, 4 soap bubbles that come together with like an X at the boundaries. You'll get kind of 2 touching each other and 2 on the sides. Very, very specific patterns that your listeners can go home and try that with their dish soap if they want. And again, these patterns are ones that just assemble themselves out of the nature of a soap film being what a soap film is. So interestingly, people have found exactly those same sorts of geometries in various arrangements of cells. For example, cells in like the eye, like the compound eye of flies. You have 4 retina cells that arrange themselves exactly as 4 soap bubbles would. Or if you have 5, they arrange themselves exactly as 5 would, and 3 as 3 would, and so on. So this and many other things just really highlight that what nature is doing, or what the fly is doing in this case, is not exactly positioning each cell in each, is not sort of manually positioning each cell in each location, but rather saying, I'm going to make these objects, soap bubbles in the kitchen's case, the cells in the fly eye case, and they, because of just the nature of their materials, will assemble themselves into some particular shape. And that assembly is very reproducible. The fly can rely on this to make a good eye. And it's self-assembly. It's intrinsic to the objects that are being assembled. Like they contain their own their own instructions.
There's something endogenous about the materials itself that is prescribing how it's going to form out into
exactly. So just the nature of the materials and the nature of the physical forces that exist between those materials. If they're electrically charged, for example, or things like that, that in itself drives them to form particular structures. So where this intersects sort of the gene's eye view is, if we went looking for what's the fly retina cell positioning gene, we wouldn't find it. And more kind of constructively, the fly doesn't need it. We don't need to specify, just like we don't need to specify where every molecule in a soap bubble goes. We just make the soap film and it makes itself into a sphere. Nature, like living things, they don't actually need to specify everything. They can specify, okay, we're going to make cells and cell membranes, and there's actually a lot of membrane-related examples of self-assembly. We can create these objects and then let the laws of physical interactions take over, and that will assemble the structure that we need. So, especially I think in a lot of popular media, there's really a sentiment that there's a gene for X, a gene for Y, a gene for Z. Everything needs a gene, but that's not really how it works. You can go a long way with just saying, I'm going to make the thing and then let sort of nature's interactions take over.
Yeah. And well, in fact, we know now from much research that there's even something as simple as height. There's just thousands of genes that are implicated, right? That's a very simple relative to a behavior or, academic achievement or something like that, in humans. And there's thousands of genes implicated, right? So even when you get to very simpler organisms, that we can kind of see the whole genome, it's still many are implicated with doing performing certain functions or actions. So regulatory circuit, right? What is this? Why did you choose this one? And why is this important? And especially how it interacts with certain types of decision making, et cetera.
Right. Yeah. So another one of these unifying principles is the notion of regulatory circuits, and this is one that I think even among biophysicists is relatively new to kind of the way we look at the living world, or really kind of internalizing the importance of this. So what I mean by this is we all know that living things, a hallmark of them is like responding to stimuli and changing behaviors and stuff like that. And it turns out, you can, so that thing of having kind of feedback from, sort of the outside going back to the inside, that's a really old notion and a very, very powerful one. You can ask, how far down does this go? And what you start to realize is that that's fundamental, even at the level of like DNA and genes, that there's kind of circuitry of like responses and feedbacks that make everything work. And coming back, actually, here again, kind of genes come up really nicely because, you can, even if you're thinking about something that is done by, is the consequence of activity from a gene, like making hemoglobin, for example, that your red blood cells use to carry around oxygen. All of your cells have the gene for making hemoglobin. But you don't want your skin cells making hemoglobin or your neurons making hemoglobin.
Right.
That activity is turned on only in the cells that are generating actually your red blood cells. So how does that regulatory activity work? Well, a lot of this works in a way that's very analogous to other like non-living circuits. You have some kind of feedback loop between kind of the output and the input. So one of the really classic examples that was actually figured out like decades ago, if you're a bacteria that wants to be digesting some sugar, or that would benefit from digesting some sugar, you want to be creating like the enzyme that'll help you digest that sugar. And that requires, reading out some gene for making that enzyme to digest that sugar. But it takes a lot of work to make an enzyme. You don't want to be doing that if there's no sugar around. So what ends up happening is, detecting that sugar activates a process that makes it more likely that gene is going to be read out. So you have kind of a feedback between the output and the input, and that lets you start, expressing these genes when you need to and not expressing them when you don't need to. So you have these kind of regulatory things, even at the level of genes, that actually tell you, when are you going to be turning these things on and when are you going to be turning them off. Where it gets really cool is you can have, in the example I just gave, you have a gene that makes an enzyme, or that is coding for an enzyme to make a sugar, and you want to detect is that sugar there. What if you had a gene whose output is turning on or off another gene? And then that gene's output or input is, turning on or off another gene. So you can make these circuits really that are kind of linked to each other. And then you can do all the kinds of things that we're used to circuits doing in a non-biological sense. They can like oscillate. You can have things go up and down and make like a clock actually out of them. You can have things have memory and like record past events. And it turns out just genes and the regulatory machinery that orchestrates them can do all of these things. So our cells can actually make clocks, and they in fact do, and like memories and all sorts of things like that. Yeah. So once you realize that you can make kind of decision-making circuitry, that again makes a lot of biology make sense. So it's not just that you have a gene for something, you have the circuits to control like when that's on, whether it's like pulsing or oscillating, or controlling your circadian rhythm, so turning on once every 24 hours or things like that.
Is this where, a lot of geneticists will become really interested with the types of gene switching that will happen? They're kind of going on and they're coming off, and they're going for certain genes as opposed to others. It sounds like there's this interplay between, you can scale this up at the, kind of at a major organism level, or even down to the cellular level of this kind of output input. And then based on that interaction, you're having certain things come online, come offline, things like that. It sounds similar to what you're saying.
Yeah, very much so. Yeah. So it is very much analogous to the macro scale things. And in fact, actually, one really kind of vibrant area in general is like, how do you fill in that gap? Like, what's the connection between the micro scale kind of feedback and regulation and like behaviors? And especially in things like flies, there's been some beautiful work, with like little flies in little arenas, sort of controlling their flight, and then looking at like what neurons are activated when they make a left turn or right turn or things like that. So really trying to link, all of these scales.
Yeah. So predictable randomness. This is an interesting one. And so how do you explain this, and kind of some of the utility here is one of the four basic principles?
Yeah. So what I mean by predictable randomness is that there's actually a lot of things, both in the living and non-living world, that are fundamentally random. So, in non-physical contexts comes up, both in very kind of deep but somewhat exotic things like quantum mechanics that have sort of an underlying randomness to them, but also in things like turbulence. You have the turbulent flow of air that's essentially kind of a random swirling of air molecules and stuff like that. But another place, so you have phenomena that are very random, but that nonetheless have some kind of overall predictability, let's say in like what their average behavior is, or what the likelihood of getting a big turbulent pulse of air relative to a small turbulent pulse of air. There's some aspect of predictability. So it turns out that that's, again, something that's true universally. So it's true in living things too. And realizing that nature actually makes use of this for how living things function is important and deep enough that I made it into one of these general principles as well. And I think probably the most important manifestation is something that is, I think, one of the most important discoveries of the last 200 years, but it's not as well known as it should be, something called Brownian motion. So most people, but not everybody, has sort of run into this at some point. If you look through a microscope at like little dust particles or little grains floating around in water, what you'll find is that those are not still. They're always jiggling around. And it was actually a botanist, Robert Brown, who discovered this in the 1800s in these little granules that were in pollen grains. And interestingly, despite being a botanist, he was he didn't just immediately conclude that this was a phenomena having to do with life, even though he was looking at pollen grains. He did some beautifully constructed experiments, um, looking at that motion in non-living things, looking at it when you do or don't have like evaporation going on, looking at it with high and low levels of light. Whatever he did, he always saw this random motion, this random jiggling around of particles.
Is that fair to say that these things are always active, right? They're always, they're not, even at the smallest bits of matter that we know, that there's always some active principle that's going on. They're not static?
They're not static, exactly. So nothing is ever static. And in fact, the smaller the things are, the more of that active motion there is.
Interesting.
So if you look and you see, a pollen grain jiggling around, if you could look at a molecule, that would be jiggling around even more. Relative to its size.
Interesting.
Or even, it's just jiggling around like even more. So the smaller things are, actually, the more active they are, the more they're jiggling around. This is also why we don't sort of routinely observe this. Like for big things, if I'm looking at, a beach ball that is so big, its random motion is actually extremely small. But small things are jiggling around.
Is it because of the size? One thing that I'm comparing this to, which maybe isn't the right comparison, is that usually the way I learned it was, for the heart rate of in mammals, right? Or animals, the smaller the faster, the larger the slower, kind of thing.
Yes.
So and maybe there's some element of scale there, but is it due to the size that it's moving so much faster, or it's just a kind of randomness to it?
No, it is. It's related to the size. Basically, what it is that the underlying cause is temperature. That what temperature actually is a measure of this, what's called thermal energy that's sort of all around us, and temperature is a measure of how much there is. And basically, everything, if it's at the same temperature, every object gets the same kick from this thermal energy. But if you're smaller, that amount of kick gives you more motion. So
the size is important there. So,
the size is very directly important. Yeah.
On the Brownian notion, you talk about there was that random motion of molecules. It's called diffusion, right? Is that kind of movement, right, that you're talking about? Is like it's some element of that it's diffuse in some ways. Yeah. Exactly.
Yeah. And it's diffuse, and that word even brings to mind connotations of randomness. Like diffuse, it's not moving in any particular direction. It's kind of randomly jiggling around. And so it's this constant motion, but it's a constant random motion.
Does this have anything to do with some of the things that we know, again, kind of at a bigger level, like what we understand with evolution and natural selection of, that we have adaptation, but then we also have some predictive elements, but we also do have randomness with evolution. We also have noise, right, which is somewhat kind of connected, right?
Yeah.
And so there's these things that are happening, and sometimes nature will select for it to have an adaptive element, but sometimes not. And so how does that kind of predictive randomness fit in with how we understand about noise?
Right. No, so that's a great question. So this notion of like randomness and like predictable randomness comes up in many, many different contexts. One of them, and I'm going to come back to this one, but one of them is the actual like physical motion of stuff, like this Brownian motion that you can look at in a microscope and see that nature makes use of quite a lot. But another, actually, like you're saying, this comes up in all kinds of things like evolution, for example. So there is just randomly, mutations or different recombinations of genetic elements. And so, if you look at the genes in organisms over time, their descendants and descendants and so on, those, if you imagine, what letter of DNA do I have at a particular point in the genome, and you watch that sort of generation to generation, that's also doing kind of a random motion in a way, not a physical motion, but going, randomly, let's say from A to T once in a while, and T to C, and so on. And in fact, it's not just a superficial analogy. Mathematically, people describe that randomness with the same mathematical forms as the actual physical Brownian motion randomness. So you have what's called a random walk in both of these things. And so if you want to think about how organisms are changing over time, it's not just everything's static, then we have like some selection, the wolves eat all the small rabbits or whatever, something like that. Underlying, that goes on as well, but constantly what's going on is this random motion of genes and things like that. And so understanding evolution, you actually have to model this random walk so to speak, of genes to see how things are changing in a group. It's actually, it gets a fancy name in a lot of evolutionary biology of a neutral model of evolution. So neutral meaning there isn't a net motion or selection, but you have kind of a diffuse, non-directed motion of your genes of a population through time.
Yeah, no, this is super instructive. Yeah. And then the fourth basic principle that you discuss is scale, right? And scaling. Obviously, some of my listeners will be aware of the great Geoffrey West, who wrote the book called Scale. I guess it's been out for 5 years now or so. And he had he's been working on this stuff for a long time. And he also comes from a physics background. And you mentioned him in the book, I think later in the book. And so how do you understand just the idea of scaling, his ideas on fractal scaling, which he's pretty big on? And, where do you kind of, converge and diverge from his kind of approach?
Yeah. So this notion of scaling, it's really looking at the question of like, how do things, whatever kind of property or behavior or whatever you're thinking about, how do they depend on things like size, like how big an organism is? And this again is something that we all kind of know exists, and it turns out we can say kind of deep things about it without too much effort. Like one example I have in the book is an observation that's been made for centuries, that bigger animals like elephants have thicker bones than smaller animals, and not just thicker because they're 5 times taller, so the bones are 5 times thicker, but disproportionately thicker. So like an animal that's twice as tall won't just have twice as wide bones, but like 3 times as wide bones, or something disproportionate like that. So you can ask why that is, and this comes back to also this idea. Is it just, can we just say the answer is genes? And I would say that's an unsatisfying answer. Like if you just say, okay, the elephant has bigger bones because it has big bone genes, and this rabbit doesn't have the big bone gene. Like, there's a recipe that nature seems to follow. I mean, that's maybe a very kind of elementary way of explaining it. But these things, there are things that through evolution and how we understand evolution now, with how organisms on the planet, unicellular and multicellular, there's a recipe that happens that is going on that says, well, this is bigger or this is smaller. There's going to be certain things that are applied here, and that works obviously with scaling. So how do we make sense of that? Of those things? So that's actually the real challenge for a lot of scaling related things. There's sort of 2 steps to it. One, we want to make, first of all, what's the observation? Like, how are things scaling? So it turns out that, like for bones, for example, at least for a lot of organisms, that with scales, it depends on the length of the bone raised to some power, some exponent, where that exponent is something bigger than 1. So if I make like I was saying before, if I make the animal twice as tall, I have to make it not just 2 times wider, but it turns out 2 to the 1.5 power, so a number that's bigger than 2 wider. So first of all, we want to make that observation. Then the challenge is, okay, can we think of a reason why that is the case? And here comes to, okay, what do kind of physical processes, how do they inform us about what sort of the scalings should be? So here, like in the bone example, the relevant things are, gravity pulling the animal down. And gravity depends on the mass of the animal, which is basically related to its volume, like how much stuff you have. Yeah. And counterbalancing that is the strength of the bone. And the strength of the bone basically depends on its cross-sectional area. And again, that's actually not something biological per se. That's true also, a beam of steel or a beam of concrete or something like that. Like the compression strength depends on like the area. So it turns out you have like this downward force of gravity that depends on like your volume, and the strength with which you can withstand it that just depends on like area. And volume and area are not the same thing. So to get those to balance out, you need to make the area even bigger than it would normally have to be to sort of equal that extra volume you're getting as you make your animal bigger. So it turns out you can understand this observation of a scaling behavior from asking, what are the physical forces that the animal in this case has to contend with. So where this gets, that's, I think, a kind of fun example, and I go into it in the book and stuff like that. Where this gets really tricky, though, is there are several examples where we've done the first part. We have observations that there's some scaling behavior going on, but it's not obvious what the underlying cause is. And this intersects. So I have a whole chapter in the book on what's called metabolic scaling, and this is the thing that you actually alluded to earlier. That basically small animals need a lot more energy relative to their size than big ones. And that's reflected also in things like heart rate. So like a mouse has a vastly faster heartbeat than an elephant. And in general, the smaller animals have fast heart rates and big ones have slow heart rates. So that's been observed for a long time. And it's reflected in things like heart rates, just how much food they need, all sorts of stuff like this. And the question is why? Or why does that relationship have the form that it does?
Why does it have the relationship it does? And in what way the things that, what you learn in, like a physio class or whatever would be, where does the role of homeostasis come into this metabolic rate to find the balance based on the size, the shape, and, behavior of how things are being scaled for the organism and or inanimate structures?
Yeah, exactly. So, it's interesting. So the chapter that I wrote on this was actually probably the most difficult to write of the entire book. And one of the main reason being that, but also I felt it was an important one to put in because this is in some sense an unfinished chapter. Like there isn't really a consensus on what is the underlying mechanism of this sort of scaling of like metabolism and heart rate and stuff like that. So there are kind of a variety of views. One from like Geoffrey West and others is that this is actually a deep thing related to the geometry of how we transport fluid and energy and all sorts of stuff like that in animal bodies. That you have blood vessels and smaller ones, or you have big blood vessels. Medium-sized ones branch off of those, smaller ones branch off of those, even smaller ones branch off of those, giving a structure known as a fractal. And if you say to yourself, well, the dominant challenge in physiology for an animal is like transporting stuff through its body.
Yeah.
The limiting factor should be like how much power does it take to push stuff through kind of a fractal network of circulatory system or whatever.
Right. Or why does this animal have blood vessels or ligaments or things like that are this long instead of this long in this part of the body, right? Those ideas to understand, you talk about the fractal structures, that this is what's needed, for this organism.
Right. So what West and others have done is said, okay, if let's say that it is this kind of fractal plumbing that's kind of the dominant thing, that would give you a scaling behavior for like heart rate and stuff like that mirrors that we see in actual animals. How that depends on size. But this is contentious because because of a variety of things. One of which is people can come up with other measures of what should be the dominant things of what sets the metabolic energy required by an organism, and that would lead to different assumptions and different kind of explanations for scaling. What's worse is that it's not completely obvious that there is a universal scaling behavior that all animals, for example, follow. Because it's a hard thing to measure. Like you can measure heart rate pretty easily. Although even there's kind of issues. Are you measuring the resting heart rate or the heart rate when you're actually doing something? And these are very different to the lifestyle of organisms. But yeah, these are sort of contentious things to measure also. So as a result, there have been literally thousands of papers on this topic of metabolic scaling and so on. And there still is not a consensus or anything really even approaching a consensus of what is the underlying explanation. So it's a little bit like, imagine when I was telling you about bones a moment ago, if I just stopped saying, okay, that we know that the bigger animals need bigger bones and we hadn't figured out the sort of gravity and beam strength argument. I feel like that's kind of where we are with metabolic scaling. We know that there's a phenomena there. there are explanations, and some of them, like, some of them are compelling and probably correct. We don't know which one, and it's a fascinating but kind of messy topic.
Yeah, my small dipping into this is like, whoa, it's like kind of, it's like walking to a party like, super late, and it's like, whoa, there's a whole lot going on here, and I've missed a whole lot. Or watching a movie 3/4 of the way through. I'm reading it and I'm like, wow, there's a lot of stuff here, but there's still a lot of things that, like you're saying, it seems very, I mean, the stuff I've read doesn't seem like anyone's very confidently saying, yes, this. There's a lot of kind of like open-ended, like, well, we still have to like figure this out or see what's going on. But it's interesting hearing you talk about it. I feel that only something like the biophysical domain could really understand and get at this. You need that physics kind of background to make sense of this. I would think you would be at a pretty big disadvantage if folks didn't understand that are studying how this works, because there's not the math there for people to kind of get at if you just do biology.
Yeah, I would agree with that. Not actually so much, I think, for the math, but because like somehow this has to be tied into physical processes of like, transport. Like, how do you transport stuff, throughout an organism, and like geometry, basically. Like, no matter what the answer ends up being, I would be pretty confident that it involves transport and geometry some way. And there's plenty of like animal physiologists and stuff like that who do think about those sorts of things. So they also have a very physical perspective. So I think that kind of perspective, whether from kind of animal physiology or physiology types of people or physics types of people, is definitely necessary. There's no way we're going to answer this by looking just at like genes or something about the molecular level.
Yeah. Okay, so I want to try and so we've kind of mapped out kind of the 4 principles. So now I want to get into some of the details here. I'll be super interested to see how you explain some of this stuff. I've asked other people on the podcast some of this stuff before. So,
cool.
So there's a few terms that many people now will talk about, and I really want to hear what you have to say about how you define it and navigate through it, so we can tackle these in whichever way makes sense to you. But okay, we have genes,
right?
And many people have a different idea of a gene, what's actually in a gene or what's composed of a gene. So there's a lot of people that still debate that thing. You can go all the way back to Richard Dawkins, selfish gene. And there's ever since then, people have been fighting about this. So genes, what are they? And then we have a few other things such as DNA, RNA, mRNA, and then proteins. And there's all these things working together in this interesting synchronicity. So how do you usually kind of explain this process and kind of separate out what this is and how we define this and all that?
Yeah. No, that's a great one. So I like to start off with proteins. Both because they're just beautiful and do remarkable things, but also because like it's a bit more well defined to think about what a protein is, and I'll say what that is in just a moment. But and then work from there to like RNA and DNA that kind of underlie where the proteins came from. So I like to start with proteins, and every protein is basically a molecule, like a kind of long chain-like molecule. And it does something. So I mentioned earlier, like hemoglobin, which binds to oxygen. There are proteins that respond to light, and you have those in your eye, and so they undergo changes when photons hit them. You have proteins that make up kind of the elastic material, your skin and stuff like that. So proteins do things. But one of the really kind of amazing and actually incredibly important things about proteins is that every single different type of protein, like hemoglobin or collagen or something like that, is fundamentally just a long chain-like molecule. So you can just imagine like a ribbon or something like that folds itself into a particular 3-dimensional shape. And this unfortunately doesn't convey in the podcast, but like I illustrate this in the book. So I have a lot of illustrations.
As a footnote here, I was really kind of surprised. Books do different things, but there's really cool illustrations in the book, in the chapters, that are kind of showing what this kind of is, or representation of this is, which was actually really helpful as you're reading it. I'm kind of seeing this stuff. Okay, yeah, I haven't seen that in a while. Oh, that makes a lot of sense. That helps. And so there's, I think, in the book, when you're talking about these kind of representations or models that have been drawn out of what this 3D kind of structure looks like.
Mm-hmm. Yeah, exactly. So, so you have very specific three-dimensional structures. So there are proteins that form themselves into like barrel-like structures. So you have like a central pore and an outside. There's proteins that form themselves into structures that kind of look like stalks with legs that actually can like move around. Those are some of my favorites. So every protein, it's doing some kind of task, but it also has some very specific three-dimensional shape to it. And these shapes are often just really beautiful to think about. And this comes back actually to this question of like this theme of self-assembly. How does the protein, or what puts the protein into some particular shape? Well, the answer is the protein itself. So every protein encodes within itself the kind of instructions for folding into a 3D shape. And I go through this in the book, but it does that because some parts of the protein might be, let's say, positively charged and some parts negatively charged, so they could come together. Some parts like water and some don't like water, so they can arrange themselves for those reasons. So the protein encodes within itself the instructions for having some particular shape. Like I write in the book, it's like you imagine like origami, but it's origami that folds itself, which is really quite beautiful. Yeah. And I should say also, maybe from a historical point of view, this actually was one of the like figuring out what these structures are was really done a lot by physicists working on like how do you get X-ray, how do you use X-rays to tell what the structure is of like molecules, and especially sort of collaborations between physicists and biologists. And this was how, especially a lot of the early information about like DNA, but also like protein structures and things like that have come about. So proteins then, are these little molecules that are also like sculptures and little self-sculpted machines. So, like I said, each one is like this ribbon-like thing that then folds itself into a shape. But where does that sequence of where does that chain come from? So then we can take a step back, and now we get into the question of like what do genes do, or what is a gene? And this is something that this I think everybody does learn in high school or something like that. That there's this sort of linkage between like DNA and RNA and proteins. But it's good to revisit it, but also then again think about it from a sort of physical perspective. So what we mean by a gene. So I'll give the somewhat incorrect answer first. But the simplest way of thinking about it, what we mean by a gene is some particular sort of sequence of DNA along our entire genome, or entire sequence of DNA, some like segment of that encodes for some particular protein. So in other words, there will be in our genome like the hemoglobin gene. So that'll be some sequence of A's, C's, T's, and G's that get read out to be turned into the sequence of amino acids that is the hemoglobin protein. So we can think about this connection between genes as kind of segments of our overall DNA package. Each of those segments that corresponds to a particular protein is a gene. So we have this linkage between the two. Where things get a bit more complicated is that it doesn't just go from DNA to protein, but first we translate that DNA code into this other molecule, RNA, which is kind of structurally similar to DNA. And then that gets read out and turned into a protein molecule. And there's a lot of really fascinating stuff along that path, but I can come back to that later. But we have this connection DNA to RNA to protein. And then this gets a bit more complicated because there are, you can read out DNA, turn it into RNA that doesn't get translated into protein. So we just are at the RNA stage, but that RNA itself can do stuff like activate or deactivate other genes, or just do other activities on its own. So there can be stretches of DNA that code for RNA, but not proteins. But most people call those genes as well. So basically, what we mean by gene is some stretch of DNA that is encoding some particular characteristic or activity. And usually that means it's encoding a protein, but it doesn't necessarily need to be.
One thing there is, you say it in the book, and I never really thought of it. And it was I remember I put the book down and I read it, and I said, I thought about it, and I was like, oh man, there's so many things here. So you say that our genome has 20,000 protein coding genes,
right?
And then the main question comes after that is, well, we have 3 billion base pairs. So what the hell are the other genes doing if they're not coding proteins? And so it was a very provocative question. And I was like, it's not like it's like, oh, well, 1.5 billion are doing this. Like, no, there's 20,000. So you have 2 billion, whatever, that aren't doing that. So what are they up to?
So what you just said is actually one of the most interesting questions that's come out of mapping genomes, not just the human genome, but all sorts of organisms. I mean, like you said, there's some fraction of our genome that's actually encoding proteins, but that's actually a really small fraction of our overall genome. Right. 99% of our DNA is not encoding proteins. And this was actually a big shock, once we realized this, from genome mapping and stuff like that. The expectation was that we would have actually way more genes than we actually do. So it turns out that, only 1% of our genome encodes for proteins. And like you said, there's about 20,000 different proteins that we make. And what's the rest doing? So as best we understand, there's multiple parts to that answer. There isn't a sort of simple answer to that, but there's multiple parts to it. One is coming back to this general theme of regulatory circuits. So there are parts of our genome that, and this we know is actually really important, they don't encode for proteins themselves, but they can influence whether the parts that encode for protein are read out. Like we said before, is a cell going to make hemoglobin or not? Things that determine that.
So they're
to give a kind of simple
ancillary genes to the ones that do it. They're kind of like helpers or supporters to the ones that actually
like regulatory sequences. So regions that influence whether genes are read out or not. So there's a lot of that, and so there will be regions typically, but not necessarily, located near genes or the actual protein coding part of genes that might be, let's say, particularly attractive to the cell's machinery that starts genes being read out. And so that'll make it more likely that gene is read out. Or they might be attractive to things that just block that machinery from forming, and so make it unlikely that gene will be read out. So it's a little bit like punctuation and stuff in a sentence, that there are things that aren't words in themselves, but influence kind of how you read out other parts of it. Except that, instead of punctuation being 1% of the characters in the book, it's like punctuation is, probably over 50% of the stuff in the book. And then, so a lot of that 99% is things like this regulatory stuff. And a lot just doesn't seem to have, at least as far as we know, any particular purpose, but just keeps, it's part of our genome, it gets duplicated. A lot of that seems to have been inserted by viral elements or things that act like viruses. And at least in humans and in animals in general, there's not much of a evolutionary disadvantage to carrying along extra DNA. So we just keep a lot of that along. And then some of it gets turned into useful things, some of it doesn't. So it's this stew of stuff.
Well, it's interesting also because it seems like there's more that we have to explore and figure out there as well. So we got proteins, we have genes, we have DNA. What were and you've kind of talked about the translation into RNA. Where does all of these things fit, and then how do we have the ion channels, which some people may be aware of, potassium, sodium, chlorine, into the protein structures, and then also you mentioned the protein structures that they have these helices and sheaths that they're
yes,
involved
yeah.
That's super important, right? So just talk about that a little bit.
Yeah. So this is actually an interesting thing about this whole notion of self-assembly. That like I said, the proteins assemble themselves into particular three-dimensional shapes. One thing that's kind of interesting is that nature seems to make use of particular motifs over and over again in these sorts of shapes. So the kind of chain-like protein molecules, like very, very often arrange themselves into little corkscrews, little helices, or like sheets where you have kind of one strand and then it kind of loops back and it's parallel to itself and then loops back and parallel to itself. So you have, it kind of looks like a sheet. So these kind of structural motifs in a way are extremely common in the overall three-dimensional shape of lots and lots, like the vast majority of proteins. So it's kind of like, and in fact, these even end up having names of like secondary structure and things like that. So it's like nature is making almost like building blocks on the way to the overall structure. Kind of like if you're building a Lego structure, it's convenient to have the actual Lego bricks and not be working with individual protein or plastic molecules. So it's like those motifs are kind of like the building blocks that are used a lot.
Yeah, along with that is this one piece on the DNA side of things where you talk about the histones, right?
Oh yeah,
and how they're packed around, or how DNA becomes packed around them, which I never heard until this part of the book, or until I was reading your book. So how is DNA packed around this almost like kind of structure itself of sorts? How does this work?
Yeah. Well, maybe before getting into that, let me take a brief step back and just point out one of just a really beautiful puzzle in a way that we have. Each of our cells has a copy of our genome, or actually two copies, but we have our genome. If you just laid it out, if you put all the DNA just from within one cell, so one copy of our genome, if you laid it out, it would be like a meter long, so like 3 feet long. All that has to go into the nucleus of a single cell. So that meter, I'm not talking about our whole body's DNA, but one cell has like a meter of DNA. And all that is shoved into a space that is about 1/1,000,000th of a meter in size. So that's just kind of amazing. And this, I often, when I'm talking, I think that this is one of my introductory examples, like in my class or something. This is a physics question. How do you shove material? How do you put this meter of stuff into a space 1/1,000,000th that size? Yeah. And you might think, oh, well, maybe that's because DNA is really squishy. And the answer is no. Actually, as molecules go, DNA is actually very stiff. It's this double helical structure that we're all familiar with. It's kind of ladder-like, and it is actually a very stiff molecule. And if you just took that DNA and kind of threw it on a tabletop, you could, if you imagined it kind of like taking yarn and tossing it on a tabletop, you would expect it to be maybe like 1/10 of a millimeter or something like that, like nowhere near like 1/1,000,000th of a meter in size. It would be much bigger. So you actually have to do some work to get the DNA to be compacted to be able to fit inside like the nucleus of cells. So the DNA has to be like squished in. So how is this going to happen? Well, there's actually different solutions from different organisms, like bacteria do it a different way than viruses. But like eukaryotic organisms like you and me and monkeys and yeast and stuff like that, wrap the DNA basically around these little spools, as you mentioned, made of what are called histone proteins. So basically 8 histone proteins together make this roughly cylindrical like little spool. And then the DNA is wrapped around those. Now here we get to, again, the idea of self-assembly. Because you might imagine, okay, again, there's some very specific thing that's sitting there wrapping the DNA around the spool. But the way these two things stick together is because DNA has a very strong negative electrical charge. And these histone spools have a very strong positive charge along kind of the walls of that cylinder. They're kind of grooved as well. So just DNA and histones together will assemble themselves into this, wrapped spool structure.
Mm.
So now instead of the DNA being sort of free, it's like wrapped around these spools. Then it turns out these spools also interact like with each other and kind of arrange themselves in kind of a fiber. It's called like this chromatin fiber. And that seems to be kind of further organized, but it's not well understood like how that is. But the upshot of all of this is that the DNA isn't sort of let free, but it's packaged into these spools. And a big part of that, how is it smushed in, is this electrical interaction that sort of is wrapping it around these little spools.
It's so interesting how the understanding the shape and structure and mechanics of DNA is really important, is what you're saying, to understanding
exactly.
how DNA works itself. This is really what you're explaining here. How is this, kind of this spool of things and how it's working, how does this break down, I guess, if you will, on neurodevelopmental and autoimmune diseases that may have flaws on the DNA packaging and not necessarily the DNA itself? Is that right?
Exactly. So this is really interesting, and this comes to something that intersects, health and disease and stuff like that. Again, especially in like popular media, there's this idea that defects in particular genes are sort of the underlying cause of various diseases, which is definitely true. If you have a defect in, let's say, a gene that encodes for some neurotransmitter or some receptor for some neurotransmitter, that will definitely have consequences, and that underlies a lot of disorders. Mm-hmm. But it turns out there's a lot of ways to mess things up. And so there are actually a lot of disorders we're realizing that are actually defects in like packaging. So if you imagine, for example, even in kind of the first step of this packaging process, just wrapping the DNA around the spools. This I illustrated in the book a bit, so but just saying it in words, there's DNA that's sort of immediately on the spools, and there's DNA kind of between the spools. Spools aren't like immediately next to each other, so they're sort of more exposed and less exposed DNA. Mm-hmm. As you might imagine, it's easier to read out the genes that are in the exposed parts. And do that, actually make the proteins that are encoded by those genes and so on. So where a gene is relative to this packaging can affect its readout. But also, if you want to express genes that are in kind of the wrapped parts, that needs to be kind of unwrapped, and there are proteins and things that kind of facilitate that. So defects in any of these things, like the packaging or the unwrapping or all of those things, can affect whether we're making neurotransmitters or whatever. So it turns out that there are quite a lot of disorders and diseases. I give like a cleft palate gene or a cleft palate disorder as like an example in the book, where there doesn't seem to be anything wrong in the actual genes that are encoding the proteins, but there seem to be things wrong with like these histone-related genes that encode proteins that interact with these histones, for example, or other aspects of packaging. So how you package the DNA can actually have big consequences for what the cells are able to actually do with it. So you can have like a neurodevelopmental disorder, for example, and have perfectly fine neurological or neural system genes, but the issue could be with just how the DNA is packaged and whether those are properly being readable or not.
Yeah, it's very fascinating because when people are trying to, knowing that can be very helpful for folks that are trying to fix those problems.
Oh, right? Definitely. Yeah.
Is this something that is like just, these folks are just condemned to that's how it's going to be? Or it's like, well, wait a minute. Now that we know a little bit of how more accurately where this is there ways, with technology and science to try and, kind of augment that and say, okay, well, how can we maybe correct for this or fix it? Maybe not, but that's hope. That's the hope, right?
Yeah, very much so. And that also is driving. So there's this area of like what controls these, especially these different levels of DNA packaging. That's a really kind of hot area at the moment. And, it has lots of motivations. One of which is what you just said, like figuring out like what are, potentially very useful targets for disease and that kind of thing. Another is early in development, development of animals and plants and whatever, you have cells that can turn into many different cell types, or at the very beginning, like every cell type. So you have so-called stem cells. And a lot of what makes a stem cell actually has to do with packaging and making things sort of more accessible, or sort of closing off different sections of the genome to be inaccessible and making others accessible, and so on. So if you can understand those, you can also potentially reprogram or sort of reinitiate stem cells and stuff like that in the lab, and sort of generate different cell types and organs and stuff like that. So this has a lot of very potentially powerful practical uses.
Yeah, no, that's something to keep looking out for. So let's talk real quick about lipids, right?
Oh, yeah.
And then we can move to the gut. So with lipids, how does the lipid bilayer architecture continue to help us understand cellular biology and multicellular organisms, and how can the membrane in there be very organized?
Yeah. So lipids, I love lipids. They were my kind of first. Well, not exactly my first, but my first sort of major foray into biophysics. I did some stuff with proteins, including ones that have trouble with this folding. That's a fun story in itself. But anyway, lipids were my first major foray into the biophysical things. And so what these are, so if you think about, if you just picture in your mind a cell, so everybody's, you're imagining some kind of blobby thing. So that has some membrane that's the outer boundary of it.
Yep.
And also, like in all of our cells, there are organelles within every cell, and those also have a membrane at their boundary. And something that's really remarkable that I think is not sort of well known is that boundary is a liquid. So it's not like a plastic bag or something. That boundary is actually like a liquid. And it's a really odd liquid. It is basically a two-dimensional liquid. So it's made up of these molecules. Well, there's proteins there too, but the main kind of architectural ingredient are these molecules called lipids. And each lipid molecule, a lot of people may have seen this long ago in school, but anyway, every lipid molecule has like a head that likes water and a tail. It's typical of two little tails that don't like water. And so what happens is they'll form themselves into a two-molecule thick thing where all those heads that like water are on the outside. So you have the heads that like water are on the outside, and then the tails that don't like water are on the inside. So you have a two-molecule thick thing where the heads are on the outside on the inside and on the inner and outer surfaces, and the tails that don't like water are on the inside. And this again is actually self-assembly at work. So every single cell membrane, made by every single cell in your body, has this fundamental architecture of this two-molecule thick sheet of lipids. But nowhere in your body or in any other organism is your cell just sort of putting lipids one by one into the structure, and sort of making the lipids. And if you just make lipids and put them in water, they will form this structure, because that's the structure that shields the tails from water. So just lipids and water will self-assemble into this two-molecule thick sheet. Just kind of amazing.
It is amazing. And just on that, is that maybe because of just our evolution? 70% of us is made of water. We come from water. Long time ago, we were in water, and then we went to land, and all of our common ancestors. So is that maybe a part of it?
That's actually a really deep question. The answer is yes, but people think it's actually even more fundamental than that. I mean, this is of course hard to test, but there's a lot of speculation that the first cells, before they were even really cells, were just sort of spontaneously formed sort of shells of lipid sheet. So, 2 molecules thick, like a bag of lipids, which is actually also very easy to just make in the lab by mixing up like lipids and water. Because these like lipids or lipid-like molecules occur naturally. In fact, they're basically soap. So they occur naturally. And so getting membranes or sort of membrane-like things to assemble themselves is actually quite easy to do. So a lot of people, myself included, think that these were, with just organic chemistry starting to occur 4 billion years ago on Earth,
right.
These kind of structures were forming all over the place. And then you sort of started just by chance to encapsulate kind of molecules that were able to kind of seed their own replication, but within this kind of protective shell of a membrane. So I think it very much ties into kind of origin of life and evolution things, likely in a very fundamental way.
Yeah, which is, you stop and think about that for a minute. It will it's a kind of a mind blowing thing when you think about it, but
it is, and it's one of the things that's also just really fun in the lab. Like I mentioned, you really can just mix together lipids and water, and they will assemble into the structure. And in my lab, actually, what we do a lot is put like fluorescent probes into this, so you can like see them. They're kind of glowing. And you just look through the microscope, and there are these, glowing shells of lipid membrane, and you didn't have to do anything except mix these together. You can't help but think about, was this going on in some tide pool 4 billion years ago?
Yeah, when you think about, the whole planet was water at one point, and then it was, well, how did things, how did we get unicellular organisms to just start to develop? And then as you get to multicellular organisms, and so you can kind of, doing experiments in a lab, you can be like, maybe there was the right kind of environment. Of course, the planet was extremely different 4 billion years ago, but there's certain things in the environment where that's maybe just kind of how it was.
Exactly.
Which goes back to your further point of, everything's connected in one way, but there's this element of these things endogenous to themselves have this kind of, I don't want to say blueprint, but this way of how they self-assemble of these these things we just do. And part of what it makes it to be that thing is kind of this self-assembly kind of piece.
Yeah. Well, I mean, I think it comes to this deep idea that you basically want to encode self-assembly into the organism's instructions, or that's what nature has figured out to do. So, which is a really kind of beautiful thing. So you're not kind of encoding the exact blueprints of the structure you want to make, but you're encoding, here's how you make a lipid, for example, or here's how you make an amino acid chain, and then you rely on these kind of universal forces, hydrophobic and hydrophilic interactions or stuff like that to do the work of actually putting it together for you. Yeah.
So before we get to actually the gut, I want to ask two things here about cells. So one thing is spatial cues, how they work and how the regulatory networks are implicated, and then lateral inhibition, right? Which is basically how cells work together and talk to each other. So how does evolution know how to do that with different species and how it all kind of plays out?
Yeah, so this is a really cool thing in general. So like spatial cues, this comes up a lot in a lot of different contexts. Like how do you know, to give just sort of two examples, you're a bacterium. How do you know to try to go, left rather than right? Not left and right, but one way versus another way. Like how do you tell one direction from another? Or if you're like a cell in a tissue in a developing organism, like you or me, how do you tell like where you are in the organism, which might determine, do you form, a foot or a hand or or another finger, or your thumb or your little finger. So how can you detect cues like this? And this, so this is important, but it also is one of these things where if you think about it a little bit, you think, wow, this is totally going to be impossible because of this randomness that's there all the while. But then if you think about it a bit more, you realize that randomness actually offers the way to make this work also. So I'll tell you what I mean by that. So let's suppose, if you're like a bacterium and you're like looking for sugars and you're trying to figure out, is there more this way rather than that way? So you're swimming around, and if you think about it just as a static snapshot, you might think, you can just tell somehow that there's more sugars, in front of me than behind me. But and that often is kind of the cartoon in a lot of sort of textbooks or something like that. Everything, we're unfortunately, I feel like we're very used to kind of static cartoons. But really what's happening is these sugar molecules they're undergoing Brownian motion. They're constantly jiggling around. The one that was in front of you, maybe behind you, like a millisecond later. Like everything is moving around a huge amount.
Mm-hmm.
So how could you possibly tell what's the good direction to go? So that's the challenge. And you think, wow, this is like impossible. But what the bacteria do, or what they've, what they've evolved to do, is not try to figure out, is there more in front of me than behind me? But instead turn this into a problem of, is there more later or now? So they will swim for some amount of time that's kind of long on molecular scales, like a second or something like that. And their sensors of, am I detecting sugars, are sort of constantly reading out, am I detecting sugars? And so if that's more now than it was a second ago, we continue going in the same direction. If it's less, then we think, oh, this is not a good direction. I'm just going to randomly, I'm going to twirl around, like a, I don't know, some random thing, like a top that's lost its balance or something. I'm just going to twirl around and pick a random direction and then do that again. So basically, if things are getting better, I keep going. If things are getting worse, I randomize. And that randomness is if it's worse, I randomize. And so randomness is built into that too. But as a result of that, I can, on average, navigate myself towards the good direction. So I can detect basically these spatial cues using this kind of a strategy. And sorry.
No, go ahead.
Yeah. So there's been a ton of work on this because it's both, a very fundamental question of how do I sense where I'm going, but this also is, if I'm a pathogen, how do I sense where my host is? How do I navigate towards cues? Or how do we disrupt processes like that if we don't want particular, like bacteria, for example, colonizing a wound or that sort of thing? So what are the mechanisms there? And I mentioned, I refer to this theme as predictable randomness. And the predictable part comes in that, okay, given all of that, you can actually mathematically lay out how good a job can I do as a bacterium trying to pick a particular direction, given on average, what amount of time I'm moving for, what the concentration of stuff is. It turns out there's a very kind of fundamental limits on what information is being is possible to extract from this kind of random information source. And I don't know if this is surprising or not surprising, but nature is very good at being pretty close to those optimal limits on what information can I get from a random source.
It's interesting because I like how when you're explaining it, you're using the explanation to illustrate further many of the things that are these four basic four principles, which is this is showing you this kind of predictable randomness and why it's important, right? Even at how cells work, intra and intercellularly. So I think that's really important.
Well, thank you. And that's something that was interesting when writing the book. That really kind of I thinking about just sort of one chapter in itself, you don't sort of think, you think about that, but especially when putting it all together, you realize that, these things really do link together across, across scales, across time, across organisms. So
so the gut microbiome, right? This is something that you've looked at. So it's almost a type of organ itself. I don't have the book in front of me, but there was a book that came out. It will come to me. That talked about, it was written by an MD and someone else. I'm drawing a blank, but there was a book basically just explaining all of the ways in which the gut microbiome is implicated in the whole body. And they also made this kind of comparison of, it basically is like an organ itself. So, how does what is it and how does it interact and why is it super essential to how we understand the human body and how does the biophysical perspective help us understand the gut microbiome?
Yeah, it's a great question. And this one, it's interesting also, now, kind of the gut microbiome is almost like a household word now. People really know.
It's in magazines now, and it's on blogs. And, people are catching on. But
I remember I was walking around just on vacation in India a couple of years ago, and there was this gigantic like yogurt ad that was like, be nice to your gut microbiome or something like that. It's like, oh, they're following me everywhere. Yeah. So, yes, it's really been amazing how much this has really caught on in people's sort of understanding of what is going on inside them. So yeah, the gut microbiome, like as I think a lot of people know now, we carry along with us this really quite enormous number of bacteria. Mm-hmm. And roughly, you have about as many bacterial cells in your body as human cells.
Right. Yeah.
Which is just kind of mind blowing.
We're the hosts, really, is what it is.
Exactly. we win on volume because the human cells are bigger, but in terms of number, the bacteria probably win by a slight margin. So if you ever held something for a vote, you would lose. So you have a lot of these bacteria, about a kilogram or so. So comparable in mass to like your heart or things like that. So it really is something to think about kind of as an organ. Mm-hmm. And the vast majority of those are in your gut. Mm-hmm. You do also have microbes on your skin and other places, but the majority of these are in your gut.
Gut meaning stomach, large, small intestine?
Correct. Primarily the small and large intestine. Not the density in the stomach is pretty low, but the intestines especially. Mm-hmm. Yeah. So you have a lot of them, and it's turning out to be the case that they influence just a huge number of your body's processes, and this isn't unique to humans either. Like sure. Animals in general, and actually plants associated with roots and stuff like that have microbial communities. So it's a very common. In fact, it is the common thing to have. So these affect things like digestion that are not terribly surprising. I mean, these are in our GI tract. They can affect how we consume stuff, but it turns out they also affect things like how our immune system is being trained to respond to different sorts of things. It affects the reasons that are still quite murky, a lot of quite complex diseases like diabetes and various cancers and so on. Mm-hmm. So there's a lot of basically feedback between this gut microbiome and a lot of your body's processes. And it really is this kind of organ level thing. But yeah, it's composed of trillions of bacteria of many, like hundreds of different species. Mm. So it's quite complex, and that's one of the things that makes it very challenging to say something about that. There's a lot of variety, and there's a lot of variation between your gut microbiome and mine are different. Mm-hmm. Mm-hmm. My gut microbiome and my gut microbiome a year ago are different, although they have a lot of overlap. But it's not even a constant thing of that. So yeah, it's this system that we're trying to make sense of, and this is something that is so it's actually now the main focus of my research group, trying to get insights into the gut microbiome, and it's something that is not commonly approached from a biophysical perspective. And we know a huge amount these days about the gut microbiome, but predominantly our information about it comes from things like DNA sequencing. So you take, for example, fecal samples and look at the DNA, sequence it and figure out like what species are present. Through various techniques, you can also learn about RNA and kind of like what genes are being expressed and that sort of thing. And that's hugely informative. And that's how we found, for example, that there are correlations between the microbiome and various diseases and stuff like that. But the reason I got into this is that those methods, they're very powerful, but in terms of figuring out what's there and what genes are present and so on. But it doesn't actually tell us anything at all about kind of the spatial organization, like the physical structure of these bacterial communities inside us. And making analogies to other organs, if you want to understand how the heart works, it is important to know that it's pumping and it has valves and vessels and stuff like that. That's kind of central to its function. So one might think that understanding kind of the physical structure of the gut microbiome is important, but also from like an ecological perspective, if you want to understand like an ecosystem, it's important to know, are all the species just mixed together, or are there some that live in the trees and some in the ground and some run around and some don't? Like, what's the interaction between these that make an overall functioning ecosystem? Right. So, like my lab's approach has been, let's actually use some really cool like microscopy and imaging techniques to try to figure out what is the actual physical organization of gut microbes. And we can't look at that in humans, but we can in zebrafish, which are like pretty transparent at young ages. So it turns out that as we kind of predicted, but as is reassuring to see, there is interesting variety in like the spatial structure of different like gut bacterial species. Like some form dense aggregates, some swim around a lot, and that seems to influence like how they colonize the gut and how they compete with others and that sort of thing.
Yeah, well, I'm remembering the book now. It's Gut Feelings: The Microbiome and Our Health, and it's it's by Alessio Fasano and Susie Flattery, and so they're medical doctors. So it's very it reads very medical. In many parts. So there are some parts that are kind of dense, actually.
Interesting.
But it was,
I haven't read that one.
Yeah, it was really good. It was really good. It's Gut Feelings. It's I think it came out earlier this year, if I'm not mistaken.
Cool.
And
I've made a note of that. Neat.
Yeah, it's a good book. It's again from a medical perspective, but I think the thing that is most arresting about this realization of the gut microbiome is how implicated it is with the brain, with the immune system, with the endocrine system, cardiovascular system, etc. And so that's why it'd be really interesting to see in your research and maybe others, how do we understand the biophysics of it? Right?
Yes.
How are these things, working throughout the body, and what are the pieces that are required or not required, and how does that? Because the assumption would be if you're trying to take an integrative, holistic approach to understanding humans or just organisms in general, this is a big piece of it that could say, where is all this stuff giving us some answers about other things in the brain or in the body, etc. So I'd be very curious to see how that advances.
Yeah, I totally agree. And I think understanding those connections between the gut and other things, is really fascinating. And as you know, that's a big topic of a lot of research these days. And I'm really excited about it.
Yeah, it's great.
Yeah, I mean, I think the big challenge is to move beyond kind of correlation to figure out like what are the causal links between all of these. That's what everybody wants, and it is really challenging.
It's hard. That's hard. It takes a long time to get to that, but I think that I think if you're having many disciplines working on
exactly right.
In tandem.
So that's actually really good. I think it really does help to have a lot of different disciplines. I'm actually part of a little center we have here at the University of Oregon on like host microbe interactions. And it's been really neat because we have some people in like neuroscience and ecology and evolution and microbiology and development and all of those things, and me from physics. And like our weekly meetings are a lot of fun.
Yeah, I'm sure. Yeah,
they're sometimes dizzying, but
that's a lot. There's a lot going on there. It's interesting when you get people. It's so fascinating where like people you'll be talking about the same topic, and it's just a whole other way of talking about it. And you're like, are we talking about the same thing? Like, and it's just different language, different words. So it's really fascinating to see that.
Especially at the beginning, it takes a lot of work just to sort of establish, what is do we have a shared vocabulary and how do we
right. And then there's interest and personalities, and so stuff takes a while.
It does,
So the last part here I want to discuss is just how we read DNA, right? And you talk about a lot of things in the back half of the book about certain types of sequencing. You talk about just what's important about DNA. One thing I kind of want to land on, if you need anything before that, I'll give you some runway to describe it. But I haven't talked about it too much here, so I just kind of want to get your ideas and your thoughts on it. Is this idea of CRISPR, which is this kind of gene editing? So maybe tell us, you could just kind of in a general way tell us about like, sequencing, how we're reading DNA, how we're doing that, and then kind of what people have been doing now with CRISPR and what that is. And then we can talk about some of the ethical landscape for it.
Cool. That sounds great. Yeah. So, as you said, pretty much the last third of the book is on kind of reading and writing DNA. So gene editing, but also like learning DNA sequences and stuff like that. And I start out actually with reading DNA. So in other words, how do we figure out what is the sequence of A's, C's, T's, and G's in our genome? And then knowing that, you can tell what proteins we encode, and there's a lot of information there. Again, this is also, I think, nowadays becoming sort of common knowledge. There's information in our genome, so how do we read that out?
Right.
It is, yeah. Anyway, so I start off with the genome, the question of how do you read DNA, and one of the things that I think is just really amazing about this, and is not well known, is that we've developed a lot of really beautiful techniques for reading out like what is the DNA sequence. So this started out with something called Sanger sequencing, which is how the first human genome, like in the 90s, was tackled. To more modern things that I'll come back to in a moment. But interestingly, like all of these actually, like fundamental to how they work is harnessing what are the physical properties of DNA. So, which is often not kind of mentioned or kind of, well, a lot of things like that are kind of glossed over in like popular media and stuff like that. But like, the classic thing of the Sanger sequencing, which is, like I said, how the first human genome was done, it relies basically on, chopping up DNA into little fragments and then applying an electric field and getting the DNA to like move through a gel. And it turns out that you couple in things with like replicating DNA that I'll skip over at least for the moment. And but really central to this is this notion that DNA is an electrically charged object, and its motion, like if you have a fragment of DNA, its motion through this gel depends on what that fragment is. So it's by kind of separating out these fragments and like reading, what are the attributes of each fragment that we're actually able to assemble, that we're able to like read the human genome at all. So if DNA weren't a long electrically charged molecule, none of this would work. And then even with the more contemporary techniques, they make use of things like, when you add a new letter onto a piece of DNA, you release like a small ion, so a small charged molecule that has electrical charge. You can use that electrical charge to like affect current flow in a transistor, in fact, and so read out what letter has been added onto your DNA. Again, things that not only like make use of things like electrical charge of DNA, but again make use of, in fact, physical instruments for like reading that out. So and then one of the very recent techniques for sequencing DNA is to actually get DNA to thread itself through a tiny little pore that's just barely wider than DNA itself. So this is something called nanopore sequencing. And on either side of that pore, you have liquid. And you're also measuring the electrical current that is going through that pore. And how much current you get depends on exactly what letter of DNA is passing through your pore. So, so you can read the sequence by measuring an electric current. So without worrying about the details, but there's this thing that writing about it really made me appreciate it, and then try to convey that all of these technologies, the fact that we can read your DNA sequence and mine and the coronavirus DNA sequence and all of these things is made possible by the fact that we understand kind of the physical properties of this material, which is, I think, rather beautiful.
Yeah, it's incredible how, is it 2000 or 2001 where we did the whole full like genome mapping or whatever it was?
So
right.
I talk about this a bit more in here, but I want to make sure I don't get the date wrong. But 2001 was the announcement of the 90% complete coverage of the human genome, and in 2003, 99%. And this was both the sort of publicly funded Human Genome Project and Craig Ventnor's sort of independently funded project kind of working together and sort of racing to do that. So that was it took many years. So it was started in the 90s and reached its completion in the early 2000s.
And to know that now where we're at with it and how we're able to sequence it and how quickly we can do it and all the technology with it is incredible just in 20 years.
It's mind-boggling. Yeah,
it is.
That the time it takes and the cost have both just plummeted. So that first human genome cost about $3 billion, billion with a B, to figure out, to sequence. And so So the cost per genome was like $1 billion. The cost for an entire genome now is less than $1,000.
Right. Yeah.
So in 20 years, we've gone from $1 billion to like less than one millionth of that.
Yeah.
Which is just, it's, I think, one of the most dramatic kind of technological advancements like ever in like human civilization. I think we still haven't really absorbed how dramatic that is.
Yeah, no. Sometimes I've said this in other contexts for other things, but I sometimes think that technology and modernity moves too fast. I think we're still sometimes we're just like really excited with the next thing, and it's like, well, wait, like what we just did was super awesome, and like we haven't had time to sit with it. Like, sometimes I find that happens. It's like 20 things happen in like 6 months. It's like, whoa, like let's just like kind of like digest some of this stuff. So that's an incredible feat. So about CRISPR. So
right, great. So
what is it, and what are some of the potentials for uses? Why are people excited about it? And we talk about some of the ethics.
Yeah, excellent. So what I was just talking about is reading DNA. So just, what is the sequence there? So the flip side of that, of course, is writing DNA. So changing what the DNA sequence is. And, there are lots and lots of reasons one might want to do this. If you have, a harmful mutation in a gene, for example, you might want to, it would be amazing if you could like repair that if you have like, a defective hemoglobin gene or something like that. So there are all these situations in which one might want to modify a genome. So in some sense, we've been doing this actually for quite a while. So we can make, actually, like as an example from my own lab, we look at, let's say, immune responses in zebrafish. And one of the tools we have to do that is zebrafish whose genomes have been edited so that they're expressing like a fluorescent protein in immune cells. So then we can look at them in the microscope and see those cells and see, where they are and where they're going and stuff like that. So we've been able to do this, but there have been kind of two big limitations for a long time. One of which is that the way, or I guess they're kind of two related limitations. You can do things that sort of randomly put genes into an organism. By randomly, I mean like its location in the genome might be random. So let's suppose I want to put this fluorescent gene into something. I'll get a fluorescent protein gene somewhere in the genome of my organism, but it might not be in a place that is easy for the organism to read out, or that it's often reading out, or stuff like that. The other limitation has been, okay, there may be techniques where you can get things more specifically located, but they have a low likelihood of working. And if I'm working with, fruit flies or something, maybe I don't care because, if 1 in 1,000 work, I just take the fruit fly in which this works and, get on with my life. Right. But if you want to try to, cure diseases in humans or do things otherwise that have, where getting it to work reliably is really crucial. That's been kind of the holy grail for a long time. So that's where CRISPR comes in. And skipping to the punchline just for a moment, basically what CRISPR is a way of editing genes very specifically. So at a specific location, doing things like either disabling them or putting in like new genetic material. So the story behind this, and here again, like the existence of CRISPR now is like, people know that there's this thing out there that's potentially very powerful, but like what it does, I think is a bit less well known. So the story behind it is actually a really fascinating one, both for the science, but also for almost what do you call it, like the sociology of science or the history of science or something like that. Because CRISPR, it turns out, depending on how you look at it, you could say it was invented, but a better way of putting it was that it was discovered. So it turns out that people, especially a researcher in Spain, and I'm always very bad with names, so I've forgotten his first name already, but a researcher in Spain made this really, Francisco Mujica, discovered this really odd thing in various like single-celled organisms, that it looked like they had repeating sequences of DNA in their genome, so like repeating sequences. And then later on, it was realized that by Mojica and actually a couple of other groups as well, and I talk about this in the book, that these repeating sequences flanked sequences of genomes from viruses. So this was really weird. Like, why on earth? Like, what's going on here? And so, two questions one could ask are, what's going on here, and who cares? So, both of which are interesting questions. And the second question is one that is a tough one. And in fact, a lot of people didn't care. And a lot of these groups actually had a lot of trouble getting funding for this kind of exploratory work. That there's just something weird going on in these single-celled organisms.
Right.
But thankfully, they were able to kind of persist with this and come to the realization that these organisms actually had a machinery, these so-called CRISPR proteins and Cas proteins, these CRISPR-associated proteins, that were able to recognize particular DNA sequences and cut them out of genomes and insert them into like their own genome. And the way these single-celled organisms like bacteria and archaea were using them, it kind of was as kind of like a library of what viruses have I encountered in the past, so I could mount like an immune response essentially to them later on. So what the bacteria had managed to figure out to do is create this CRISPR system that finds specific DNA sequences and cuts them. So then a couple of people, especially Jennifer Doudna and Emmanuel Charpentier, but also, again, I'm terrible with names. And this I unfortunately will not pronounce properly. Sorry about that. But Virginia Siskinis in Lithuania. Anyway, several people started realizing, hey, we can actually like take this system and program it not to cut these sequences that the bacteria wants to cut, but whatever sequence we want to cut. And the reason is that what CRISPR uses as its guide to where in the genome do I want to make a cut is basically a sequence of RNA. And if you can replace that with whatever sequence of RNA you want, your CRISPR protein will cut where it finds kind of the match to that RNA in a genome it's found. So what they all managed to do is this really beautiful thing of then taking these proteins, adding in your own external guide, and these CRISPR-Cas system will then cut DNA at whatever target site you've given it. So you've made a way to cut DNA. So that in itself, you might think, okay, cutting DNA doesn't seem all that useful. But your cells don't leave cut DNA alone. That's detected as DNA damage. And so your cells will repair the broken DNA. And two things can happen when you do that. If they just repair it, odds are pretty good that it's not a perfect repair, and so you'll miss part of the sequence, and so you'll deactivate that gene that the cut was located in. Right. So you've turned off that gene, basically. Or if you have just extra fragments of DNA that you've intentionally put in, it can splice those in as it's doing the repair, and you've now made an insertion into the genome. So in a nutshell, that's CRISPR. There's a lot of ways of kind of making this kind of more robust and modifying it and stuff like that. But basically, you're harnessing this naturally derived protein, this CRISPR-Cas system, that has figured out this biotechnological challenge of finding a specific DNA sequence and manipulating it. And you're then using that to either deactivate or insert a gene.
So the problem with this, well, there's a lot of advantages.
There's a variety of problems. Yeah,
there's advantages, but there's problems. The first advantage that people would say is that you could essentially early on in I don't know if it's in utero or whatever, but if you can find the genes that are potentially would be implicated for autism, Down syndrome, people even says with Alzheimer's, things like that, you just take them out and replace them with better genes, right? Is the idea, at least,
right?
And people have then extrapolated out from that, like, well, what if I just don't want blue eyes, I want brown eyes, or if I want straight hair, not wavy hair, and we just take out the gene for that, or genes for that. And this is where it starts to get very ethically
Yes.
problematic in some ways.
Right.
So how, in your estimation, for what you know about this, where is the kind of, or is there an ethical compass here where people are like, well, wait a minute, how do we do this, right? We can do research and we can see the potential and look, we can actually do this,
right.
But who's on that committee saying, well, you can't do it for this, you got to do it this time, and how is those conversations happening?
Yeah, so that's a both fascinating and super important question, actually, and it's one that I think people realize that it's an important question, but I think it is still discussed less than it perhaps should be.
Yes. Yes.
So there's a lot to say about that, but I think a couple of key things that I think are important and that I also tried to convey in the book. One of them is, I think it's impossible to talk about those questions in a constructive way unless one has a good grasp of what do we mean by genes even, and like what these different machineries can do.
Right.
And here, I think there is a lot of kind of popular misunderstanding of genes that makes, like, to be a bit more concrete, something like, actually an example you brought up earlier, something like height. There isn't a single gene that encodes for height, right? There are thousands that influence height. In fact, I talk about that actually in one of the chapters of the book as well. So one of the things that I think people worry about isn't as likely as a lot of people who worry about it think. In other words, let's suppose I did want to somehow, use CRISPR to make a taller child. Right. That would be a terrible idea. It would be stupendously difficult to do because there isn't a single gene there that one would be editing. And it's, I think, in any foreseeable future, I'd be willing to bet that even for a century, editing 3,000 genes is just not going to be like a tractable thing to do. So I think a lot of the kind of complex and beautiful, aspects of humanity are not single gene things. So you're not going to be kind of turning on or off a gene for them. So the use case, I think, really is much more the situations that are one gene is doing something bad, and those are overwhelmingly like disease related. Like here's the gene that, the cystic fibrosis transporter gene or whatever,
or maybe
that
ALS,
is a gene.
I don't know.
Or ALS,
that's
a good,
you're right.
But I have, if I can repair the cystic fibrosis transporter gene, that's a single gene that repairing that will do something. Those are the situations that are vastly more realistic to imagine. So I think grasping that will kind of change the framework a little bit of how people think about things, and it actually might change the framework of which issues people think are more important to discuss. Because there's a whole bunch, and I'm not sure. I sometimes feel like one of the more important issues that is sometimes discussed, but is particularly important is, let's suppose we do say, okay, it is worthwhile to fix a cystic fibrosis gene. Is that kind of ethically acceptable? Is it accessible to people, or is this are things like this things that will be, especially if they're not covered in public health and things like that, are they only accessible to the well-off? Those I feel are even more important discussions. There was something else to point out. Oh, right. Another interesting thing is, as you may know, right now, the vast majority of countries and researchers agree that doing any CRISPR editing of like human embryos, actually like embryos that go to term and are develop into babies and so on, is not allowed. It's not a good thing. There is a Chinese researcher who did it, which has shocked lots and lots of people, including the Chinese government, who clamped down on this and pointed out we're not doing this anymore. So at least at the moment, there is this widespread agreement that we should wait on any human embryo CRISPR editing. But it's, I think, not obvious what the next step is. Like, what are the criteria we're going to use? Because, the technology will only get easier and easier. And there will be people who quite legitimately say this could save this could save a life or something
Yeah.
like that.
Yeah.
It's also interesting, I think, that I don't know where I fall on a lot of this, but there's some very interesting historical parallels with things like in vitro fertilization,
Mm-hmm.
which is nowadays quite common.
Right.
I forget what the fractions are. I have it in the book somewhere, but
Mm-hmm.
it's quite common. But there were a lot of people who were very strongly opposed to that when it was first developed and felt this is playing God, this is unnatural.
Mm-hmm.
And coming back to what you were saying before of like kind of rapid changes, it's kind of shocking how quickly we're, as a society, we think, that's perfectly fine.
It's just another option.
Exactly,
Why would
it's
you
just
do it?
another
Right.
option. Yeah.
Right.
So I don't know enough to make a prediction, but I'm really curious if if something similar will happen here.
Yeah. It's very interesting. It's something that I don't spend a lot of time thinking of, but every now and then it will pop up, and I'll like, where are we going with that? So your comments were really helpful. Last question I have for you is, what is your first book written for a popular audience? Folks that have listened to this, I think will be really intrigued and interested, right? Because of the way in which you're describing these things and talking about, really complicated stuff. What is the things that you want people that read your book, what do you want them to take away from? Like, what's the impact that you hope to achieve with that?
Yeah, that's a great question. I think more than anything, I want people, I really love nature.
Mm-hmm.
I like going for hikes. I like, seeing animals. I like watching nature documentaries and all kinds of stuff like that.
Mm-hmm.
And I really do feel like understanding the kind of biophysical perspective, it gives you kind of a whole nother way of appreciating nature.
And like, the lens. There's a whole
lens.
Yeah,
a whole different lens. Yeah. You realize, okay, the lion and the squirrel, they're very different, but they have this underlying similarity that, they're all like us using DNA and proteins and self-assembling lipids for their architecture. There's this like universality to stuff.
Right.
And, the wildebeest and like the deer you see, munching on your garden or whatever, all have the same issues about like bone strength and mechanics. Like there's this kind of deep, unifying principles that do actually like tie things together in the world. And I tried to provide a lot of examples with that. And I feel like if I've helped anybody kind of realize, nature is even more beautiful than I thought. That's one of my main goals.
Yeah. That's wonderful to say.
As a secondary goal, because I do encounter a lot of people who quite, and this is a perfectly fine view, but really want to ask, what's the practical use of all of this? Sure. There is also the fact that, a lot of things, like I've said with like DNA sequencing and stuff like that, really are made possible by taking seriously the physical nature of living stuff. So it's useful too. But if I had to say one thing, I do try to convey kind of the underlying beauty of everything being tied together.
No, yeah, that's wonderful. The book is called So Simple a Beginning: How 4 Physical Principles Shape Our Living World. Raghuveer, this is a great conversation. Tell folks where they can find your research, where they can get your book and find you online or anything like that.
Oh, yeah. So the book, So Simple a Beginning, it's from Princeton University Press, and it's popular audience trade book. You can search for it on the Princeton University Press site. In addition, I have a blog. It's called The 18th Elephant. So if you Google that, you'll find my blog, and I write kind of random things on it, including, if you search for the book title, you'll find my posts on that and a couple of like representative illustrations, stuff like that. If you want to find my research website, the easiest way is to probably just Google my name and the University of Oregon. But it's probably also, I think it's also linked to on my blog's about page, and you should also feel free, your listeners can feel free to contact me also. I'm happy to get emails, and that information is on the blog as well.
No, that's wonderful. Thank you, so much for coming on and talking to us about all these wonderful things, and for really making it, accessible for folks. This is a wonderful conversation. So I'm really glad we had it.
Thanks a lot. Yeah, your questions are great, and it's been a lot of fun.
All right. Thank you.
Thanks.